So Professor Barker gave you an introduction on the central dogma, or actually, no, I think I did by video lecture. Okay. We made it through. Here I am in person. What we're going to be doing over this next quarter is diving in on each one of those individual components of the central dogma through this quarter. And then one thing that was not added to that lecture but is added to this semester is we're going to have an additional brand new lecture right at the beginning of quarter 4, which for lack of a better title we're calling Extra Dogma. That's going to talk about all sorts of other things related to gene and gene regulation that doesn't directly involve replication, transcription, or translation. Today and into next class, we'll be talking about replication. And in contrast to some of the other lectures that you've seen in this course so far that have been wrapped around various diseases, you don't really have the opportunity to do that for replication because no DNA replication generally means no viability. And so there's no diseases that are intimately tied to defects in DNA replication. Instead, what we're going to talk about are the man-made ills of chemical and biological warfare, of which we have a long history, whether we're talking about the Spartans with their pitch and sulfur, the Sung dynasty with their arsenical smoke, or the Mongols catapulting plague-contaminated corpses. This has a long history in our society, and we're going to talk about 2 more modern innovations, if you will, in warfare, and we'll circle back about how they directly impinge upon DNA replication as we make it through this slide deck. First one is purely chemical. It's mustard gas. Full name chemically is shown here on the left. What it does from a chemical perspective is that it serves as an alkylating agent. What is an alkylating agent? This is a chemical compound that adds alkyl chains to other molecules. Mustard gas chemical structure is shown here. The means by which it serves as an alkylating agent is through these 2 alkyl chains here. The chloride is a good leaving group. If a nucleophile like a nitrogen, of which there are many nitrogens in biomolecules, attack this site here, you get the alkylation, the release of the chloride, and these are the basis of the chemical modifications that this compound executes. It was originally discovered in the 1800s as a chemical warfare agent. One of its major mechanisms of action is as a vesicant. Vesicant is a blistering agent. The chemical structure is simple. That was why it's been around for centuries. Easy to make, easy to disperse, very stable, hard to treat, and decontaminated. Used extensively in World War I before they outlawed chemical warfare internationally. Yes, why isn't it not mayonnaise gas, right? Yeah. I will talk about that, I think, in the next slide. Yeah. So some of the symptoms, right, they're pretty gruesome. Erythema. This is red, and you see with the eyelids there, redness and irritation, inflammation on the mucous membranes. All right, so that's one of the really bad symptoms. Conjunctivitis. Inflammation, also eyelids, eyes, and things. Erythema refers more to mucous membranes in general. I touched on its ability to serve as a vesicant, so there's the blistering and the burning. Where the mustard comes in is how it smells. So the nasal irritation, sneezing, nosebleeds, mucous membranes in your nasal passages. I have read it smells like mustard, horseradish, garlic kind of combination. As the exposure progresses, you get coughing, sore throat, again, mucous membranes, vomiting, and then the LD50, this is the lethal dose that kills half the population that receives this exposure, is only about 1.5 grams worth of the compound, okay? And if it reaches in the lungs, only about 10 milligrams is required to kill the person. And you see all of these blistering effects at hundreds of milligram quantities over a certain duration, depending on your surface area, because it affects disproportionately the epidermis and the keratinocytes in your skin, and this is what gives rise to the blistering. We're going to touch on how mustard gas elicits some of its effects from the perspective of DNA replication and DNA damage. Mustard gas is going to be the chemical warfare agent. Our bio-warfare agent is anthrax. Anthrax has a long history, not necessarily in bio-warfare, but from its name, a synonym called the wool sorter's disease. The anthrax is derived from a common soil-dwelling bacterium called Bacillus anthracis that either dwells in the soil or infects large animals like sheep. One of the characteristics of anthrax that is advantageous from a weapons perspective is that it forms spores. This is a dormant form of the bacterium that's highly resistant to adverse environments. So when the bacterium sporulates and goes into its sporulated form, it can persist very long periods of time, no water, high humidity, low humidity, heat, cold, and then can begin replicating again when conditions are more favorable. So that's one of the reasons why it's a favorite for weapons makers, not hard to grow, easy to disperse. I'll touch more on the mortality of it in a slide or two. The other one that is advantageous is that it's non-transmissible. If dispersed properly, it can infect the person who it was dispersed to, but it doesn't give rise to an outbreak of anthrax. It's not contagious. So you deliver to the one individual and it doesn't go any further. So for targeted interventions, I suppose, it's advantageous. Where anthrax got the name of the wool sorter's disease is that the most common normal way to contract anthrax is through the cutaneous form. You have a wool sorter who's shearing the sheep, cuts their knuckle, and then gets the spores, the bacteria, into their skin. What this gives rise to is a bump here. Here's a photo of it on the right-hand side. You're sort of numb to the pain, but that gray, yucky section in there, that's a necrotic center. I believe Professor Barker talked about apoptosis as a form of programmed cell death. Necrosis, some people call cell murder. All right? So these are cells that die in an unregulated way, release all of their components locally in the vicinity of where the death occurred, highly inflammatory, and is characterized by this kind of black and grayish appearance. And then there's also an immune response, lymph nodes, secondary lymphoid organs. I talked about that before. And later on, we'll touch on how the innate immune system plays very prominently in the response to aerosolized anthrax, which is the very lethally dangerous form. So the question is, what is special about the spore? From a biological perspective, I presume. Okay, so this would be, if you want to talk about a bacterium that's actively replicating, okay, so it's making DNA, all the things that we're going to be talking about now, making proteins, making RNA, dividing, growing, okay? The spore is, if you will, a form of hibernation, okay? So it goes into there's usually a morphogenesis that occurs with the spore. It's going to change its shape. It may have a protective shell around it, and nothing happens until the conditions are more favorable. There'll be an environmental trigger that causes that spore to reactivate and then go back into its proliferative state. And so it's a state change that over evolutionary time is advantageous, because if there's a drought, if there's a flood, if you happen to encounter highly acidic conditions or alkaline conditions, those spores can persist, whereas the actively proliferating bacterium cannot. Yes, that's a good question. So if you talk about infected animals, where is the infection? Cutaneous also. That's why if you're shearing the sheep, you're right in that vicinity. Okay, so it's also on the skin of the animals. Inhalation anthrax, totally different story compared to cutaneous anthrax. This begins by delivery. Okay, this bacterium doesn't ordinarily fly. All right, you have to aerosolize it. If it is aerosolized and somebody were to inhale it, now the infection starts not on the skin but in the lungs. There's an incubation period. We'll speak more about that later on, probably tomorrow. But then after that incubation period, there is a series of symptoms here of appearance of an upper respiratory infection that there's nothing to write home about. But then it quickly goes into shortness of breath, profuse sweating, diaphoresis. You heard about dyspnea before. Myalgia can be muscle pain. And then septic shock, this is the latest stage, massive, overwhelming systemic infection giving rise to loss of blood pressure in the individual and then death. This happens 100% of the time within 1 to 2 days of these symptoms going. So it's very irreversible. There's only a limited number of examples of people being able to intervene in time. And we're going to talk about this antimicrobial Cipro later on about how it's effective at treating these anthrax, biowarfare, aerosolized anthrax in individuals. And I show the lethal dose there for completeness. Sorry, what is diaphoresis? Profuse sweating, the inability to control perspiration. This seems like a lecture on apples and oranges, right? I mean, this is very different agents, but they both converge upon aspects of DNA replication. We're going to speak about how mustard gas lethality and the cellular damage that it causes is partly due to the damage it elicits on DNA itself. And then that antimicrobial that I said was effective against anthrax infections is its mechanism of action is by inhibiting bacterial DNA replication specifically, and we'll speak more about that at the end of this lecture. Let's begin with a quick review on a slide there wasn't an official pre-lecture for this class, but in the central dogma one there was, and this slide appeared. We're going to be talking about DNA, base pairing, and the two strands that make up DNA. So to remind, we have two DNA strands come together to make our genome. They're held together by hydrogen bonding that occurs between A's and T's, adenines and thymines, G's and C's, guanines, cytosines. They're schematized here. A's and T's come together with 2 hydrogen bonds. G's and C's come together with 3 hydrogen bonds. And the orientation of those bases and the hydrogen bonding patterns that they can accomplish is what achieves the specificity, the exquisite specificity of DNA as a means for replication. Right? The back and forth in the hydrogen bonding and this base pairing is referred to more generally as complementarity. And so A to the T, that's the complementary base pair. G, C is the complementary base pair. And when you have these complementary strands bound together to one another, they do so in an anti-parallel manner. There's a polarity with nucleic acids, just like there's a polarity with proteins. N terminus to C terminus with DNA and RNA, it's 5' to 3'. And you'll see why it's 5' to 3', right, in today or next time. So we have the 3' end of one strand and the 5' end binding to, hybridizing to is another term we might use, to the 5' end at 3' end of the opposite strand. When a cell undergoes replication, what occurs is that each of those strands is used as a template to create another copy. Because you have this exquisite complementarity among the different bases, all of the information that you need to replicate the double strand you get with a single strand. And the enzyme that performs that replication and the polymerization of DNA is called DNA polymerase. It's an enzyme. You've learned about enzymes before. All right, so here's an enzyme that creates DNA polymers from nucleotides, nucleoside triphosphates, formally. For DNA replication to work, there are 3 major challenges that DNA polymerase or the whole replication machinery must overcome to have this be effective for the DNA polymerase enzymatic activity to achieve DNA replication in cells. And so DNA polymerase itself has a couple of problems. We're going to talk about all the really neat and interesting things that DNA polymerase does, but it also has a couple of handicaps. It can't do everything on its own. First is that DNA polymerase cannot start the replication process at a couple of different levels. To start DNA replication, you need to be able to expose the single strands that are going to act as the template for replication. But in cells, they're already bound to one another. They're wound up in that double helix, right? And DNA polymerase cannot get in there by itself. We need to separate those strands or melt them, okay, to be able to expose the bases that will serve as the templates for replication. Second problem is that DNA polymerase can only extend a pre-existing strand. In other words, if you have a parent strand, you have a strand that's serving as a template, and then on top of it, you already have a little piece of DNA bound to it, or nucleic acid bound to it. It can take it from there and extend all the way through, fill in the gaps, but it can't start it from the beginning. So we have a starting problem with DNA replication that needs to be resolved. And then the third problem is that DNA polymerase, when it catalyzes the polymerization reaction, the direction in which DNA synthesis occurs only occurs in one direction, 5' to 3'. That's why the convention is 5' to 3', because it's in the direction of DNA synthesis catalyzed by DNA polymerase. But I told you already that the double-stranded DNA is anti-parallel with one another. So we have a directional issue where if replication is proceeding up or down, depending on how you look at things, you're going 5' to 3' on one strand, but 3' to 5' on the other strand as you move in those directions. So this creates another challenge that we'll get to. There was a question. Yes, and so I think it was in the video recorded lecture when we talked about an RNA primer to start, that's the motivation. That can be used as the starting template for polymerase to go forward. We'll talk on how that primer gets laid down and how the strands separate in a couple of slides.
So why can it only go 5?
Why does it only go one direction versus another? Well, it'll show some crystal structures of the enzyme with DNA in it as a substrate, as well as the nucleoside triphosphates that are the monomers that are being polymerized. This is a massive, massive enzyme. There are geometric constraints on the way you have to fit the double helix or the parent strand in, and so it's a massive geometric coordination to get everything just right. And then as you zoom down to the molecular level, you have to position the phosphates and the nascent strand that's being extended just right. To whip it around and go in the opposite direction, it's almost, in many ways, you'd have to start from scratch to be able to evolve an enzyme in that way, because everything's like the mirror image of one another. You'd have to do it backwards. Yeah, so the question here is about this directionality. And so if let's say we have, this will be the template strand for this example. So let's say that this is a piece of DNA on top here that's being replicated 5' to 3'. DNA polymerase is going in this direction. The synthesis occurs. That's the direction in which the synthesis occurs. So you can get turned around a little bit by what your frame of mind is. But just remember this, that the synthesis occurs 5' to 3'. We've taught these lectures for a number of years, but this is one example where we did a recent update. And so I think I've only done this one or two times. The revisions that we put into this lecture is to try to give a little bit more of a human comparison here. And DNA replication is really remarkable in the sense that you would think that it would be this deeply conserved process that like happened once ever in evolution, and then that was it. Not so much. There's a lot deeper conservation with translation in the ribosome than there is in DNA replication. So the way that the next couple slides are going to arrive to you is a compare and contrast between prokaryotic DNA replication and eukaryotic, or we'll say human, right, DNA replication. There are certain steps where the themes are broadly similar, but the details are profoundly different. And so I would like you to appreciate the similarities and the differences. But it's really akin to something like flight, right? So flight got invented 3 times: insects, bats, and birds. Okay, so it's like a convergent evolution type of thing with DNA replication. So some of these slides, where we begin is where does DNA replication begin, right? And where DNA replication begins is at what's called imaginatively an origin of replication. So we talk about replication origins, origins of replication, right? This is a site on the DNA that is going to be replicated that serves as the starting point for replication. Recognized by origin binding proteins. We'll speak about what those origin binding proteins are in just a moment. Where prokaryotes and eukaryotes diverge is on what defines the origin of replication. In bacteria, there are certain short repeats that can be defined at the sequence level, called consensus sequences. More to come on that in a second, that define and allow you to predict where origins of replication are. In us, it's nowhere near as clear. However, what's common between both the bacterial origins and mammalian origins is that they usually have a rich stretch of AT base pairing. Yes? Are the consensus sequences the same as motifs? Yes. From an informatics standpoint, it largely, the difference in the terminology arises from if you're talking about DNA or proteins. So proteins tend to have motifs, and DNA tends to have consensus sequences. And that's just because they're different fields and people call things differently. But the idea of recurrent features is the same, and I'll talk about that in a moment. Can anyone wager why AT characteristic would make sense from the perspective of an origin of replication? Bond's easier to break. 2 versus 3, actually 33% easier, okay, compared to G and C. All right, that makes a big difference when you're talking about the molecular level. And so this is the reason biophysically, thermodynamically, why you need to have these AT-rich stretches, but the precise orientation is not very well defined in humans. Consensus sequence, I can elaborate a little bit on the question before about motifs. How do you define these consensus sequence? Well, the way that you do it is you look at a lot of origins of replication. So let's say that this one is from anthrax. Let's say this one is from E. coli. This one is from subtilis, blah, 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 all the way on down the line. All right? You line them all up. You slide them left and right to one another to try to see if there are any common features. You can do this on a computer. And then what you're looking for are recurrent sequences of A's, T's, C's, and G's that come up again and again and again with a similar spacing from one organism to the next, from one origin to the next. And if you can identify these consistent features, then this defines a consensus sequence. So I'm showing these for origins of replication, but there will be others when we start talking about sequences for regulation of transcription and things on down the line from there. Maybe I'm going to say this here. From this point forward, the left, the 5' to 3' convention is buried in every display of the DNA. All right, so the 5' starts at A, G, G, T, A, A. So you see from the upper left-hand one? I don't need to specify it. Some more things about replication orders. Well, what happens if a cell, be it bacterial or human, is going to replicate its DNA, and there's an origin of replication? What occurs? Left-hand side is prokaryotes, bacteria, okay, with a single circular genome. And on the right-hand side are, let's say, mammals. Chromosomes, linear. That's why it's a long line across on the right-hand side. Both have origins of replication. Typically, bacteria only have one. We have many ones, and as I said, they're less well defined. And so what happens is that during replication, you're going to have replication initiate at the origin, one place on a circular genome, various places along a chromosome. And then once that origin has had the two strands separated, more to come on how that occurs at the biochemical level in a moment, we create what's called a replication fork. And this is the separation of those two strands. Now you have the two parent strands pulled apart from one another, and then here's the fork. All right, so there's a fork on the left, a fork on the right, both proceeding off of that origin of replication. Similar theme in mammalian cells, two strands, you have two parents. They get separated locally. They also generate a replication forks at the end, but because there are many decorated along the chromosome, they're also called replication bubbles because they kind of all the way around on the chromosome. Once you establish that replication fork, you have DNA replication happening here and also happening here. It runs all the way around the genome until it reaches the end. They get separated from one another, and now you've replicated a prokaryotic genome. Same theme with mammalian genomes. These replication bubbles expand as more daughter DNA is synthesized. They eventually fuse together with one another. And then once all of these replication bubbles have fully fused, you have the two replicated daughter strands, each one base paired with a parent strand. Next series of slides is going to walk through step by step which each one of these things involves, just going from the beginning at the origin of replication through to the replication of the DNA. Yeah, so your question is, you have the origin, you create the fork, talked about how there's going to be 5' and 3' character on both of those forks, all right? And so 5' to 3' DNA synthesis is occurring on both of those replication forks in exactly the same way, but flipped from one another. Okay, so, can you envision that geometrically? So whatever's happening up on the upper replication fork in my hands here is occurring on the lower replication fork on the lower strand on the other replication fork, and vice versa for these two. I'm trying to dance around the more detailed explanation because we have about 15 or 20 slides before I talk about that. But you get the idea of the topology we're working with here. Okay, step by step through each one of these. There's an origin. We're going to initiate DNA replication. What do we need? We need an initiator protein. Easy, right? Initiator proteins bind to origins of replication. And they may be able to initiate the strand separation that I said is the beginning point. Here's where we have some of the divergence. What's shown here, it was really this really great science review that came out last year that prompted me to reconfigure all of these slides, because they did a great job doing a structural comparison between many of these key intermediates for DNA replication. This review was only possible because some of these structures only came out a couple of years ago. And so even though we knew that these proteins were important, we didn't have the detailed molecular information that has just come out within the last couple of years. So the two initiator proteins that I'd like you to know is the major one in DNA, in bacteria world, prokaryote world, and it's called the DNA A. DNA A is in fact what's shown in a cartoon form here. DNA A binds origins of replications, and it does strand separation. In us, we have what's called an origin of replication complex called ORC, which is a multi-protein assembly of 6 subunits here, each one color-coded, ORC 1 through 6, forming this hexameric complex that binds to origins of replication. ORC does not separate the strands. What it does do is create a location on the genome to recruit other proteins that do separate the strands. But from the perspective of recognizing origins of replication, same. So we now have an origin recognized by DNA A or ORC. What happens next? Even beyond the initial strand separation, there needs to be more unwinding of the DNA to enable DNA polymerase to come in and begin elongating, or even to initiate the laying down of any nucleic acids in the vicinity of those two strands. That further separation of the DNA is catalyzed by another family of enzymes called helicases. Helicases expend energy to pull apart the two strands, to melt the DNA. And here again is another example of convergent evolution with very different strategies. In prokaryotes, the helicase is called the DNA B. It's easy, right? DNA A, DNA B is the next step. Crystal structure, ribbon diagram is shown on the left-hand side. And us, by contrast, we have another hexameric complex with some accessory proteins here called the mini chromosome maintenance complex, which you don't need to know what that is, but you do need to be aware of the acronym MCM. Okay, so MCM, the MCM complex refers to this hexameric assembly, and it's what helps to unwind the DNA early during our DNA replication, and then also serves as the helicase that unwinds the two strands as replication occurs. Structurally, they're very different from one another. And even beyond that, they work on opposite sides of the track. All right, so DNA B, it does its unwinding by assembling its complex on the strand here that moves in the 5' to 3' direction. So it's unwinding by binding to the one strand and then moving in that moving as shown on this schematic. The MCM complex also binds to a strand, but it moves 3' to 5', so it's bound to the other strand and as it moves. So this is more akin to the question before, like, why don't you just work in the opposite direction? You can, but you need a lot of evolutionary time to be able to do it, and you can see the strategy for the two is completely different at a molecular level. But the net result of the two is the same: DNA unwinding, creating real estate for DNA polymerase to extend the daughter strands off the template. When the initiator proteins bind, when the helicases separate, as that replication fork moves along, think about this in your mind. If the helicases are going along with the replication fork, behind it, all right, if the parent strands are not immediately replicated, thermodynamically, those two strands are just going to want to hybridize back to one another. They're complementary, they're in the immediate vicinity. It's thermodynamically unfavorable for them to be separated. And this is what they will do within seconds. Unless you have these things called single-stranded binding proteins, or SSBs. Right? So these are abundant proteins that are in the nucleus or in the chromosomal, the genomic material of prokaryotes that stabilize single-stranded DNA in the single-stranded state. You can think of them as almost like coding proteins that keep those two individual strands apart, such that the DNA replication and elongation can occur before they bind back up from one another. And then the last thing that you need, and this is where all of my props behind here are going to come into play, is that we need one more enzyme to resolve a topologic problem of DNA replication caused by the separation of a double-stranded right-hand helix. Right? These things are coiled around one another. And if you take the two strands and you separate, you're supercoiling the two strands on the other ends as you're pulling them apart. And that creates enough mechanical tension that can do DNA damage, can stop replication things. And so the way that thermodynamic barrier is overcome is through a family of enzymes called the gyrases. What gyrases do is tackle the supercoiling issue that I'm going to show schematically in just a moment by breaking and remaking breaking and rejoining together the DNA to relieve the supercoiling. Gyrase is one specific example of a broader family of enzymes that relieve issues with supercoiling that are called topoisomerases. There are two types of topoisomerases. The first one, which is pretty straightforward, are called type 1 topoisomerases. Regrettably, type 1 topoisomerases are not the most involved ones in DNA replication, but these come into play with gene regulation, so transcription, and also chromatin condensation and things mitosis. So when you got to untangle chromosomes and you got to separate them apart when the cells divide, this is where these type 1 topoisomerases come into play. What do? Well, they can recognize regions of high supercoiling. They bind to the DNA, and they cut once on one of the strands. And then they're going to unravel a little bit of the DNA to relieve that supercoiling tension, and then they reform the backbone right where they nicked it the first time. Because the nicking has some energy associated with it that's stored in the topoisomerase, and then it gets put back in when you reform the bond, there isn't any net energy expenditure for this type of mechanism of action. Right? Crystallographically, this is what a type 1 topoisomerase looks like. Here's the template supercoiled DNA in the cleft here. Type 2 topoisomerases, gyrase is one example of. Type 2 topoisomerases recognize really tangled pieces of DNA where if it's so coiled on top of one another, it's going to wrap around in the vicinity here. And what type 2 topoisomerases will do is cut both strands, two of them, okay, not a single strand, and then pass the extra loop through the place where it just cut, and then form the loop on the back end that was originally broken by the enzyme. This cartoon doesn't help very much. All right, I don't understand what this meant either. But gyrase is a type 2 topoisomerase, and then it's at this point I'm going to demonstrate this, but I need 1, 2, 3, 4, 5, 6 volunteers. Come on up. Okay. All right, you guys first. You're going to be the DNA ends, so can you stick this on yourself? DNA end, stick it on yourself. Come over on the other side here. Two DNA ends, DNA ends. Pull them all out. Okay, now make them DNA. You can help. Or you can slow him down. You have to coordinate. And if you can help me try to line these two up. There we go. Did they do it right? Hold on, did you do it right or what? Anybody? Why not? Lucky guess, but if you'll take it, right? Okay. Good. Okay, so this is our DNA template. We're going to go in the mammalian world here, so I need somebody to be orc. Somebody be orc. Come underneath. Thank you. And then let's go right about there. Just bind it. Yeah, like that hexameric complex. Okay, so then that's where it's gonna happen. MCM underneath. all that happens here in the mammalian world is a handoff. All right, once the origin has been recognized by ORC and it recruits MCM, this is not a direct recruitment. There's some accessory proteins in the middle there that you don't need to know, but it's not a direct binding between these two. We can maybe demonstrate this as a handoff. So hand off that origin to MCM, okay, and then you can go and sit down, or you can move over to the side there. But from the biological perspective, the org is no longer required after that origin binding event. All right. You're the helicase, okay? So start melting the DNA. And then we're going to need help from single-stranded binding proteins on both strands. All right, so if you can separate those two. Oh, actually, take off your label. We need another helicase because we need 2 forks. So I don't have another tag. I did this kind of last minute. So separate out these 2 directions. Cool. And then you guys hold them apart here. Turn around so everybody else can see on the other side. Hold it up like in the cartoons. Come on. Thank you. Keep going. And then I'm going to sneak behind you. Oops. We good? Is it getting to be a bit of a mess there? The DNA ends, you're being too kind. Yeah, keep going. Well, don't go past this. Here, pull this tighter. Keep going. And here, loosen it up a little bit. I'm just pushing it. Hold on, wait. See this? Okay, that's the supercoiling that I'm referring to. All right, so I'm going to first take the role of the type 1 topoisomerase. Not relevant for DNA replication, but I just want to get across the idea of what it does. All right, so what a type 1 topoisomerase would do is take these 2 pieces, unwind them as follows. Okay, I still need you to keep that fork open. Yeah, okay, and then relax the coiling. Okay, so relevant for DNA condensation, relevant for transcription if there are some tangles in the DNA, but not for DNA replication. So I'm going to try to wrap it up again here. I have all my strands in order. So now I'm gyrase. Can you give me a hand with this here? Just hold those out. Okay, so what's happening here now is that I'm going to pass it through like this. I'm not unwinding, but you see how it's reducing the supercoiling? All right, so the net result of the looping here, you see? I got 2 turns. There was 5 or 6 turns in here, but a couple of these loops, now I've relaxed it. Gyrase brings them back together, okay? And then now you've surmounted the topological challenge of DNA replication. All right, let's give everyone a round of applause. I lose 1 foot per year. Now that we have some ideas about the geometry of how gyrase is working, I can show this also in crystallographic detail. Here is a gyrase. Here is the strand that is supercoiled that is going to get broken. Gets broken by the gyrase. The other DNA molecule that gets passed through it goes through like so into another binding pocket, and then the 2 strands come together just like I showed in the cartoon. Gyrase activity is important for those replication forks. The other place where it comes up very prominently is in prokaryotic genomes. I showed the cartoon in the beginning. I said, oh, you have the 1 origin, the replication forks, and then voila, the parent and then the daughter genomes arise. Not always. Actually, what happens is when you have that replication, they're stuck together, okay, like the Olympic rings, like this. And so here's another place where you need to have an enzyme that can cut, pass through, and then reform. Gyrase is also involved in that step for prokaryotic genomes, and also long chromosomes that are heavily tangled, similar type of thing. All good with initiation, replication complex? All right, so we've resolved the melting of the DNA. So there's enough real estate now, stably enforced by the single-stranded binding proteins to allow extension to occur. Let's speak more about the DNA polymerases. As I touched on briefly, these off of a DNA template will grow a DNA polymer from the 5' end to the 3' end. So growth is 5' to 3'. It seems each year I can never say that enough. All right, you must know this. For DNA polymerase to work, I already said this, it needs a template. That's the code that is going to get replicated. It also needs an energy source and a monomer source to be able to proceed. And that energy and monomer source is provided by deoxyribonucleoside triphosphates, or just DNTPs for short. And then, as I said, it needs to already have some sort of pre-existing nucleic acid template to elongate. And do people know the priming? Have anyone heard of this? The phrase priming the pump? We are generations apart. Okay, so then I can't even ask you about what the hell does priming the pump mean. Okay, there are these things called water pumps, okay, that you pump and water comes out. When you have to prime the pump, you need to put water into the pump machinery to create a secure enough seal to be able to pump water. So you need water to pump water. Thus, you need to prime the pump. And similarly, you need DNA for DNA polymerase to continue to make more DNA. That's the reason why it's called a primer. So if you have the primer, and you have DNTPs, and you have DNA polymerase, you can extend as follows. The DNTPs will come in when there's appropriate base pairing here with the strand as it extends. What occurs is that this 3' free hydroxyl on the 3' OH group of the strand that's being extended will attack the oxygen here, will attack the alpha phosphate on the DNTP, release the 2 terminal phosphates. It's called inorganic pyrophosphate. It's inorganic because there's no carbon, pyrophosphate because there's 2 of them there. That is thermodynamically favorable. Okay, so this is the delta G, negative, all that stuff that you learned earlier in the class. So that's the release of the energy that then will create this bond. So the need for the primer, from evolutionary standpoint, motivates the need for a primase to be able to lay down some type of primer according to rules that DNA polymerase is constrained by. And what sets the primer up is another enzyme called primase. What is primase? Primase is not a DNA polymerase, but an RNA polymerase. And it's a special kind of RNA polymerase that's DNA dependent. And what does that mean? That means that its template is DNA, but what it's laying down is complementary RNA rather than DNA. Okay, but there's base pairing. We talk about ribonucleic acids rather than deoxyribonucleic acids. A lot of the bases are similar. The pairing is similar. And for the purposes of priming the pump, it's sufficient because they're not going to stay there permanently. They just need to get a nucleic acid started for DNA polymerase to continue on. And RNA polymerases, like you'll see in transcription, don't have that priming requirement like DNA polymerases do. So they can start wherever, given a template. That's how the RNA polymerases work. There was something in the back? Resolved? Okay. And so the ability of primase to lay down that first little bit of RNA primer, in principle, will resolve the priming issue of DNA polymerase. It doesn't have the same requirement. It can start a template to get elongated, and then it's off to the races. Let's speak more about the DNA polymerase. The naming convention is different between bacteria and us. In prokaryotes, the DNA polymerase is called DNA polymerase 3 or Pol 3. In us, it's Pol delta. These are massive enzymes. They do a really important job in the cell. All right, so multiple subunits here. And we've been harping on the polymerase activity. So polymerase, 5' to 3', 5' to 3'. You do need to know that, right? But it does other things also. One of the really important characteristics of DNA polymerase for replication is a 3' to 5' exonuclease activity. I'm going to do the order of these bullets differently. What the heck is an exonuclease? An exonuclease is an enzyme that cuts nucleic acids, polynucleic acids, at the ends. So if you can envision a long polymer, 5' to 3', an exonuclease is going to whittle away bases on one end or the other. And in the case of DNA polymerase, it is whittling away at the 3' end in the 5' direction. If there are exonucleases, there are also endonucleases. Endonucleases cut in the middle. We use them for recombinant DNA technology, molecular biology. Bacteria use them to protect against phages, viruses, and things. Not relevant for DNA replication, but just be aware. Exo, outside, endo, inside. Nuclease, enough said. Why would a polymerase have an exonuclease activity? You want to go forward, right? Onward and upward, go down the strand. Except when we show the cartoon forms of DNA replication, we just say, oh, there's the base pairing, and it comes in, and the strand elongates, and off it goes. In reality, there's nucleotides all over the place, and they're all sampling into that register wherever the open end is for the strand that's elongating. Errors can be made. This is where the 3' to 5' exonuclease activity is valuable, because if an incorrect base is incorporated into the daughter strand, you're not going to have perfect base pairing with the parent. So the end result is that base is going to be stuck out in some way off of the template. As a result of that base being stuck out irregularly off of the template, the polymerase can't go forward because the 3' hydroxyl of that base is not optimally positioned to do that nucleophilic attack on the next NTP. So the polymerase stalls. And if that were the end of it, well, you have two scenarios. One, the polymerase can just stall and the replication stops forever, and that's not good. Or the polymerase just barrels on through that misincorporated base, and now you have mutations. Okay? And if these are too significant, you're going to kill the organism. So the 3' to 5' exonuclease activity serves as a proofreading activity of the polymerase. During that pause, if it pauses for too long, the exonuclease activity kicks in, it whittles off that base that just was incorporated, and then it allows a new nucleotide to be able to come in, sample the environment, and in principle get elongated. This proofreading activity massively improves the fidelity of DNA replication. If you take this away, then these polymerases will barrel on through, and I don't know, off the top of my head, I'd say you're going from error rates in the order of thousands to 10 to thousands bases without the DNA proofreading activity to millions or tens of millions or perhaps even more proofreading activity of misincorporations with the proofreading activity of polymerase. And I think this is the first time that we have this term here. We talked about anti-parallel strands and things. As this replication is occurring, compared to the parent strand that is getting replicated, the daughter strand has a different sequence, right? It's complementary to the parent strand, but it's also in the reverse order, because to remind you, always read 5' to 3'. Therefore, the daughter strand is what's called the reverse complement of the parent strand. In other words, I would read the parent strand here, CTTACAG, 5' to 3', always. The daughter strand is the reverse complement, and it would read CTGTAAG. We good with that? Another ribbon diagram, this is what DNA polymerase looks like. You can see the two strands. Here's the parent strand, the elongating strand, and you can starting to form the double helix on the way out. I touched on the fact that the DNA polymerase is a multi-subunit enzyme. There are subunits of it that are critical for that elongation activity. Another thing, if you do the thought experiment for this, is if you did enzymes and substrates, right? Michaelis-Menten kinetics, all the way back to what, lecture 2, something like that in the class. What has to happen in a Michaelian enzyme reaction? Enzyme binds to substrate. They form enzyme-substrate complex, K-cat, right? You create product, and then you release enzyme. Start the thing all over again. That would be awful for DNA polymerase if every single time it incorporated nucleic acid into the elongating strand, if it unbound, had to do a diffusive search, rebind onto the template, incorporate another base, and then it unbinds and goes back around. Okay? The way that DNA polymerases avoid that problem is that they are highly processive enzymes. And the way that they obtain or have that processivity function is what's called a processivity clamp, which is literally a complex that wraps around the parent strand and doesn't let go. And so even though you may have unbinding or the enzyme might not be optimally positioned right at the place to put in the next base, the enzyme itself never lets go of the parent strand until it has no more elongation to go. It runs off the rail, so to speak, or it hits into something that it can't go through. And so these are the examples of the clamp loaders of prokaryotes. This is another example of convergent evolution. They look similar-ish. They are repeating units here. They have a hexameric-like structure, but in prokaryotes, it's a dimer of these 2 beta subunits. And then in us, it's a molecule called pCNA, and it's a trimer. So you divide 6 by either 2 or you divide 6 by 3. You end up with the same thing, something that latches down on the DNA and then doesn't let go. Okay. So we have the processive proofreading polymerase on the replication fork. I should take a step back here, because here where what the cartoon is showing here is the helicase, gyrase, relieving the supercoiling, melting the DNA, single-stranded binding proteins here stabilizing them, and now primase has come in and laid down that RNA primer. DNA polymerase, Pol delta or Pol 3, will extend off of that RNA primer into the replication fork. On it goes, on it goes, on it goes down from here. So again, look at the orientation, 5' to 3' synthesis. Everything is good for that strand, right? 5' to 3', straight shot all the way through. You have a problem on the other strand, and this is the one that I was talking about before. You can't just have RNA polymerase be up here and then go in this direction, because now you're synthesizing DNA 3' to 5' rather than 5' to 3'. And so what occurs is that the other strand is being synthesized away from the replication fork. There is RNA primer that's being laid down. There is DNA polymerase elongating that primed strand, but it moves away from the fork. And so what this creates is an asymmetry in each one of those replication forks, where you have one strand that's being synthesized continuously. That, as you might imagine, occurs a lot faster, and therefore it is called the leading strand on each replication fork. You have if you have a leading strand, you have to have a lagging strand. So what characterizes the laggard, all right, the lagging strand, is that each one, it's getting synthesized in little bits and pieces. These bits and pieces are called Okazaki fragments by, you can imagine who discovered them. And these are characteristic of the lagging strand. I think I also want to show a cartoon of this on the chalkboard, because there's always a question after class. Let me take one of these replication bubbles. '5', 3 prime, 3 prime, 5 prime. All right. So at each one of these strands, if you have, you're starting in here, go in like this. All right, I have to keep my 5 prime, 3 prime. On this strand, on this part of the replication fork, it's the lagging strand. But as you come to the other replication fork, 5 prime, 3 prime, it's actually the leading strand. So you're going to have a lagging strand in this schematic here and up here, and then leading strands on the flipped orientation on the other side of the bubble. Very easy to get yourself turned around, the topology here. All right, and then the last thing that I want to touch on here, do I have another color? I do have another color. Remember that for each one of these, it's initiated with an RNA primer. So I draw this like DNA, but in reality, there's a little snippet of RNA in here, which is, you're not going to use, it's not stably heritable in this form. And so after this all gets filled in, so the polymerase will go in, it hits the RNA primer here, and then Pol delta or Pol 3 falls off. It can't go any further because there's a roadblock there. What occurs is that another polymerase comes in, called either Pol 1, polymerase 1 in prokaryotes, or polymerase alpha in eukaryotes, that resolves these junctions here, both on the lagging strand as well as places where the leading strand finishes or the replication bubbles come together. So Pol 1 or Pol alpha has many characteristics that are similar to Pol 3 or Pol delta in that it is a 5' to 3' polymerase. It has proofreading activity in the 3' to 5' direction. But the key added feature of Pol 1 or Pol alpha is that it also has 5' to 3' exonuclease activity. That enzymatic activity is spatially separate from these other enzymatic activities that are earlier in on this slide. And so what happens is that Pol 1 or Pol alpha comes in here and is going to be incorporating bases as it's removing the RNA primer, all the way up until it hits the next strand of DNA, no more RNA primer. So the nick there, which is where you hit it, ran into the RNA primer, moves along with each one of those strands until you see the next DNA strand there, until the primer is totally cleared out. This is a lot more complicated. Consequently, the processivity and the kinetics of Pol 1 or Pol alpha are slower than Pol 3. And so I'll conclude with a cartoon version of what we have so far. RNA primer laid down, elongation by Pol delta, right, Pol 3. There's another, let's call this another lagging strand gets synthesized behind it, runs right into the RNA primer. Then Pol 1, Pol alpha will come in, bind here, elongate, and remove the RNA primer that's in front of it, and take it all the way up to the end here. The cartoon is a little bit misleading because it actually can't bring together the final two pieces. So as you come to the very end here, you've removed all the primer, but you still have a space, and that last space is filled by a final enzyme called the DNA ligase. You call it pastase. Well, don't call it pastase, but if you remind them what it is, that's what it does. It brings two ends together, glues them together, recreates the phosphate backbone between them, and fills in the gaps between the Okazaki fragments or any other places, even on the leading strand where they came together. So to put the whole thing together, sequence of events in a minute. Initiator protein binds to the replication of origin. That could be DNA A or ORC. You have a helicase come in. That could be DNA B or MCM. Unwind. All right. The unwinding of the DNA, stabilized by single-stranded binding proteins. Supercoiling is relieved by the type 2 topoisomerase gyrase. And then in that replication fork, we have RNA primer being laid down by primase, that it is extended by DNA polymerase 3 or Pol delta. And then the RNA primers are removed by Pol 1 or Pol alpha. And then the final step is the last little nick there is repaired by DNA ligase. Repeat that all the way through the genome, the chromosome, and then you have a fully replicated strand. This is the perfect place to stop. Thanks for your attention, and then I'll see you on next class. Quiz is on all this.